So today we are going to discuss about the histology of nervous tissue, and we know that what is the function of nervous tissue. As we all know already that the transmission of electrical signals from sensory receptors to effectors means suppose anything heat or hot utensils, if you are touching, you are just moving away from that location. So this is what, this is transmission of electrical signal from sensory receptors, means skin is having sensory receptors to effectors means towards the brain. What is the location of nervous tissue? It is located brain nervous, that is the central nervous system. The spinal cord is also central nervous system. The nerves, which is located in the peripheral nervous system. After that, what is the description? What is the main components are the brain and spinal cord and nerves. We can see here that in this photograph, nuclei of supporting cells are visible here. This is the cell body of the neurons, and these are the neuron process, which contains two types of cells. First is the neuron, that is excitatory cells, then supporting cells, that is the neuroglial cells. So means what? Suppose brain, spinal cord, these are the central nervous system, which includes two type of cells. First is the neuron, which form it, and next is the supporting cells, which are providing support. Next thing, nervous system has three major function. What are the major function? First is the sensory, means monitor internal and external environment through presence of receptors. So what is sensory is doing? It is monitoring the internal and external environment through the presence of receptors. Means we have receptors in our skin, in eyes. We have receptors like cells, specific type of cells are there. So what is happening actually in this case, the internal receptors and external receptors are there on the body, and external receptors are on the environment, which is influencing light, like light, sunlight, no? So what is going on with the help of with the effect of these changes in the environment, we have to change our uh, response. So this is called through the receptors, which is found in the brain. Now integration means interpretation of the sensory information, means information processing and complex higher order functions, and motor receptors and their response to for information processed through the stimulation of the effectors. So muscle contraction and glandular secretion are the we can see these are the two responses, that is in to information proceed through the stimulator of the effectors, that is muscle contraction and glandular secretion. Then general organization of the nervous system. The two anatomical divisions are there: central nervous system, which includes brain and spinal cord, and then peripheral nervous system, which includes all the neuronal tissue outside the central nervous system. Then afferent division, that is the sensory input. Then efferent division, that is motor output. So peripheral nervous system involves all the neural tissue outside central nervous system. Okay, all the neural tissue outside central nervous system. Then afferent division means sensory input, means the nerves which is bringing the uh, we can say that that stimulation or whatever is the receptors is getting in, that is coming in the we can say that through the afferent division or sensory input. Then effector division means motor output. After analysis of the how the we have to respond and animals have to respond accordingly, the motor output or different division will respond. Then somatic nervous system and autonomic nervous system are the two parts. So we will see it in detail, because somatic means the body is responding and in our wish, and then autonomic means the internal organs are responding by their own. Now we have to see the next thing. What is the histology of the nervous tissue or neuronal tissue? There are two types of neuronal cells or neural cells in the system. First is the neurons, and second is the neuroglia. So neurons are for the processing and transfer and storage of information. So neurons are for processing, transfer and storage of information. Then neuroglia for support, regulation, and protection of the neurons. So these are the two type of cells. First is the neuron for a processing, transfer, and storage of information. That is general type of structure of neuron which we have seen. Now neuroglial cells are the for the support, regulation, and protection of the neurons. These are the another type of cells along with that found in the nervous system. So with this we can see here the neuron structure. What is visible here? This is the cell body of the neuron. These are the dendrites. These are the dendrites in this location. This is the axon, and this is the synaptic terminal. So we can see now that dendrites that is stimulated by the environmental change or the activities of the other cells. Dendritic spines are there, and dendrites are stimulated by the environmental change or the activities of other cells. Then cell body. Cell body contains the nucleus and mitochondria. So we can see here this is the cell body which contains the nucleus, mitochondria, ribosomes, and other organelle and inclusions. Then axon conduct nerve impulse or action potential towards the synaptic terminal. Like this, we can see this is the synaptic terminal, and this is having the affect the another neuron, or effector organ or muscle or gland. So like this, they are proceeding the impulse from this way to this way. Next point, we can see another photograph here which we can see having the dendrites like this. These are the synapse, means what is happening. One neuron is connected with another neuron. This is the central nervous system, this is the peripheral nervous system. So you can see that the nasal granules are the found in the cell body, that is the ribosome, along with that granule like structure is there. Then we can see the nucleus with the nucleolus is side mid position. Then axon hillock is there. Then myelin sheath formed by an oligodendrocyte. So this is the myelin sheath which is formed by the oligodendrocyte. This is the nodes of Ranvier. So these are the nodes of Ranvier which is found. Then you can see the axon. Like this, we can see the nucleus of the Schwann cell which is found around the axon. This is the collateral branches, myelin sheath produced by the Schwann cell. Like this, we can see the motor end plate at skeletal muscle. So these are the muscle, because the connection where the nerve terminal is joining the muscle or skeletal muscle fiber, that is called motor end plate, and then terminal branching is visible like this. So central and peripheral component of the motor neuron. So we can see like this. This is the from here to here, this is the part of the peripheral nervous system, and this is the part of the central nervous system of the motor neuron, which we can divide it like this. Then classification of the neurons. So neurons को भी classify किया गया है. How it is classified? Structural classification based on the number of process coming off the cell body. We can see here anaxonic neuron. Then bipolar neuron, means this is the cell body, on both sides axon is there, and both sides we can find the this terminal synaptic terminals and dendrites. Then pseudo unipolar. Okay, so this is the pseudo unipolar. We can see the axon on both sides, and initial segment dendrites is this side, and synaptic terminal is this side. What is happening here? This is bipolar, and this is pseudo unipolar, and then multipolar. Multipolar means we can see that the axon is there like this, and this is the dendrites. Okay, so this is the normal type which we have studied earlier. If the cell body is having both side axon, then it is called pseudo unipolar, because it is appearing like a single, but it is having both side axon. And this is the bipolar. Okay, bipolar means we have to see here that dendrites are in this way present, and this way the synaptic terminal is there. Then anaxonic neuron means no axon. So anaxonic neuron, no anatomical clues to determine axon from the dendrites. Function is not clearly known. Then bipolar, we can see the two process coming of the cell body. One dendrites and one is axon. Okay, so this side is the dendrites and this side is the axon. only found in the eye, ear, and nose. Bipolar neurons are found on the sensory organs, eye, ear, and nose. These are the most important three sensory organs of the facial region. So bipolar neuron, remember, is found in the this eye, ear, and nose. Next is the pseudo unipolar, means single process coming of cell body. Okay, cell body is this one, the single process. Actually, this is a single process giving rise to dendrites. Okay, so at one end, an axon making up rest of the process. So like this, we have to see that pseudo unipolar neuron is found. Now multipolar neuron. So multipolar neuron is we can see that multiple dendrites are there and a single axon, and it is the most common type of the neuron. Next point. So receive stimuli and transmit impulse. How it is possible? We have to see that the nucleus pale staining vesicular is appearing, and chromatin is fine. Next point, the nucleus is very prominent in synaptic ganglia. If centric, often binucleate nucleus is there, we can see here. Then cytoplasm, chromophil substance, and nasal granules, and neuroplasm is there. So we have to see that in this case, this nasal bodies means what, irregular clumps of endoplasmic reticulum, basophilic in nature. So these are the nasal bodies, and absent in the axon and axon hillock, are larger in motor neuron, indicate high metabolic activity, synthesize neurotransmitter protein, and chromatolysis occurs after the damage of the axon. Then inclusion, two kinds of pigments, melanin, dark brown pigment, lipofuscin, yellowish brown or orange pigment is there. Now dendrites or telodendria. So terminal axon is there. This is the nucleus. This is the dendrites. Okay. So axon or axon cylinder or axon hillock divide of needles, which we can see here. Length fraction of the 2 millimeter we can see here, and several feet long it may be there. Then collateral axon, axioplasm or axiolemma, the swan cell, that is myelin sheath forming. This is the nodes of Ranvier. Now in this photograph we can see that the nerve myelinated of the pig, which is showing in this photograph, and this is a Masson's trichome staining showing the collagen fiber also. So 2 is showing the epineurium, like which we have seen in the muscle here also. Endoneurium, perineurium, and then epineurium. Endoneurium means nerve fiber, covering of nerve fiber. Then numerous nerve fiber combinedly form bundle, and that covered another connective tissue that is called perineurium. After that, numerous type of these fasciculi combinedly connect externally covered by the another covering that is called epineurium. So we can see like this epineurium. First is the adipose tissue, 10th is the perineurium. This is the perineurium, which is the covering of the bundles of numerous, which is visible here. Okay, so similarly, which we can see in this case, similar we have been seen in the muscle, like this we can see here also. So what is visible here? 9 is showing the nasal granules in the cell body of the neuron in the histological photograph. 12 is showing you the stellate cells. 8th is showing you the neuron cell body. This one is the cell body like this. Okay, 9th is the nasal granules in the cell body. 7 is showing you the nerve fiber like this in the section it appears. Then 12 is showing the stellate cells. So dorsal root ganglia of the dog is showing this with the luxol fast blue and crystal violet staining. Next point in the section we can see here, just to take an idea that marginal plexus of the esophagus of pig is showing in this photograph. Eighth is the showing the neuron cell body, and third is showing you the muscularis externa or inner circular layer. 13 is showing the submucosa. 5 is showing the muscularis mucosa. So in this way we can see like this. Then third is showing the muscularis externa, inner circular layer. 11 is showing the plexus, and 8th is showing you the neuron cell body like this, and 4th is showing the muscularis externa in this way. So iris plexus is visible jejunum of the dogs. These parasympathetic plexus are located between the inner circular and outer longitudinal layer of the muscularis externa of the digestive tract. So in the jejunum, iris plexus is showing in this photograph, which is having parasympathetic plexus are located in between the inner circular and outer longitudinal layer of the muscularis externa of digestive tract. So inner circular layer, outer longitudinal layer, this is the showing the iris plexus, which is having the this is the plexus, earth is having the cell bodies. So in this way it is appearing in the cross section, just have a idea. And another photograph is showing you the this is the axon in the section, then endoneurium is the second one, which is showing you like this. Then I told you the covering of the nerve fibers in the innermost lining is called covering is called endoneurium. Then third is showing that this is the myelin sheath, and then fourth is showing you the perineurium. This is the perineurium is covering of numerous nerve bundle which is having endoneurium. After that, similarly, bundles are again involved in a covering that called epineurium, as we have seen. Now, nerve fibers, if you know that, we already discussed that nerve fiber is having what? Axon and sheath. So, in peripheral nervous system, axon covered with thin protoplasmic sheath, that is called neurolemma or sheath of Schwann. That is this one. Provides medullary sheath myelin to axon. Then in central nervous system, myelin is provided by oligodendroglia. So in the central nervous system, this myelin is covered, it's covering, that is provided by the oligodendroglia or myelin wrapping the over the axon. That is called oligodendroglia, which is covering in the central nervous system. Means inside the brain, the nerves are having provided along the axon, that is myelin, which is provided by, which is by formed by the oligodendroglia. While in the peripheral nervous system, means outside the central nervous system, means outside the brain and spinal cord, the nerve axon are covered with protoplasmic sheath, that is called sheath of Schwann or Schwann cell, and provides medullary sheath, that is myelin to axon. So we can see that the Schwann cell surrounded all the axons in the neurons in the peripheral nervous system, creating a neurilemma around them. And neurilemma allow for potential regeneration of the damaged axon. So it is this neurilemma having the potential to regeneration of the damaged neuron. This kind of possibility is there. Now, stellate cells support the group of cells bodies of the neurons within the. So, stellate cells are what? They are supporting the group of cell body of the neuron within the ganglia of the peripheral nervous system. So, that is the cell of the peripheral nervous system. So, in this photograph, we can say Schwann cells are visible. These are the axons, and covering is called Schwann cells. Okay, so this is the myelin covering internode. This is the Schwann cell nucleus. Outside it appearing like this, so we have to analyze accordingly. And these are the nodes. So what is the difference myelinated axon and unmyelinated axon? Myelinated in the peripheral nervous system, as we have discussed here, that which is provided by the Schwann cells, but in the central nervous system, means brain and spinal cord, it is provided by the oligodendroglia. Now connected, we can see that the endoneurium is sheath of Henle, it is also called, that is the delicate tube of reticular fiber, which we can see here. Then perineurium, a dense connective tissue sheath surrounding the bundle of fiber. And then epineurium, loose connective tissue sheath surrounding the peripheral nerve. Okay, so epineurium is this one, this one is the perineurium, and below that, inside the fiber around the outer covering is there, that is called endoneurium. Then blood vessel, endoneurium contain capillary network fibers, as we can see that the A, B and C, means thick high velocity conduction fiber A, B is medium, and C is thin and no myelin or low velocity conduction fibers are there. Now anatomical structure of the neuron. So these are the blood vessels we can see in the neuron. These are the fascicles. These are the endoneurium. This is the Schwann cell of the myelinated axons. This is the perineurium around one fascicle. Then, like this, we have seen in the muscular tissue. Similarly, here also, the endoneurium is the covering of the fiber, that is nerve fiber. Then perineurium around the fascicle, and then we can see all over the covering is called epineurium. That is the outermost covering. Similarly, in between them, we can see the blood vessels also. Now, function, after that, we have to see that classification of the neuron. Functional classification based on the type of information and direction of information transmission. So, sensory neuron, motor neuron, and association neuron. Sensory or afferent neuron means what? That transmits sensory information from receptor of peripheral nervous system towards the central nervous system. Suppose if we are touching anything that is hot, so in the peripheral nervous system, because we are, suppose we are touching with hands, so in these fingers are having the opening of the nerve terminals of the sensory type of neurons. So that is received, that is that part is called peripheral nervous system, and that signal, or this we can say that that is going and analyzed in the brain. So this is called transmission of sensory information from receptors. So these are the receptors which is found on the skin, and that is called receptors of peripheral nervous system, towards that is going to the brain and spinal cord at the central nervous system. So most sensory neurons are unipolar, and a few are bipolar. Next, motor or efferent neuron. So transmit information, that is the transmit information from the central nervous system to the effectors, means muscle, gland, and adipose tissue. So transmit information, motor information from central, means brain is transmitting to the effectors. And what are the effectors? Muscle is there to contract, to move accordingly. Then gland is there to provide secretion accordingly. Then adipose tissue is there, accordingly they have to respond or move in periphery of the body, and all are multipolar. So these are the motor neurons are the multipolar maximum. Next, association or interneuron, that transmit information between neurons within the central nervous system, analyze input, coordinate output, and are most common and type of the neuron. They are 20 billion, and all are multipolar. Okay. Next, come to the next, that is the nerve termination. As we can see in this photograph, that the motor end plate is visible, means foot like nerve terminal on the muscle fiber. Endoneurium becomes continuous with the endomysium and sole of motor end plate and many muscle nuclei. So we can see like this, they are contracting and forming the motor end plate. These are the nerve terminals of the uh, this neuron, and they are innermost covering is called endoneurium, which is in contact with the endomysium, means covering of the muscle fiber or myofibrils. So that is why this kind of because you know that if any sensory information is coming with the help of peripheral nervous system, then it will analyze in the brain with, and then we accordingly the response is given by the animal. So what will happen? This nerve terminal will give the response accordingly to the motor end plate, and motor end plate is having the muscle, so muscle is contracting accordingly. Now receptors, so it exteroreceptors, external stimuli like touch, pain, pressure, smell, sight, and hearing. Then proprioceptors, stimuli within the body wall, movement and posture. Then visceroreceptors, stimuli within the viscera. So all receptors, depending upon their terminal arborization, come under two classes: free non-capsulated nerve and encapsulated nerve. Then free non-capsulated nerve, these are found practically in all epithelium, in connective tissue, in muscle and serous membrane. And here the fibers, after losing their myelin, form a subepithelial plexus from which the axon and their branches between cell and end in knob-like terminal swelling. Then encapsulated nerve endings. They include end bulbs, Meissner capsules, Pacinian capsules, muscle spindles, and muscle tendal spindles. So these are the encapsulated nerve endings. So what we have seen that receptors are three types. That is exteroceptors, that is the external stimuli like touch, pain, pressure, smell, sight, and hearing. Then proprioceptor stimuli within the body ball, that is the movement and posture. Then visceroceptors stimuli within the viscera. And all receptors, these exteroceptors, proprioceptors, and visceroreceptors, depending upon their terminal arborization, comes under two classes. Okay, so we can divide them according to their terminal arborization as two type. First is the free or non-capsulated nerve or encapsulated nerve, and then that is the uh, non-capsulated and encapsulated. So free or non-capsulated means that is naked nerve endings. These are found practically in all epithelia in connective tissue, in muscle and serous membrane. And here the fiber after losing their myelin form a subepithelial plexus because they are naked nerve. So there is a losing of the myelin form subepithelial means just below the epithelium. That plexus is forming from which the axon and their branches between cell and end in the knob-like terminal in swelling. This is the thing here happening. Now encapsulated nerve ending, they include what? That is the encapsulated, that is not naked, that is the capsulation is there. So this is end bulb, Meissner capsules, Pacinian capsules, muscle spindles, and muscle tendon spindles. These are the encapsulated type. Now one by one we have to see that encapsulated type of endings. So first is the end bulb or Krauss end bulb. That is one type of encapsulated or capsulated type of receptor that is spherical or oval in shape and consists of thick lamellated capsule of connective tissue cells and fiber surrounding a central cavity or inner knob. Then within this end bulb, the naked axon of one or more myelinated fiber and either simply or in the form of skin. Then end bulb are found in the lip, tongue of man, cheeks, soft palate, epiglottis, nasal cavity, lower end of the rectum, peritoneum. Okay, then other serous membrane, tendons and ligaments, and connective tissue of the nerve trunks, synovial membrane of certain joints, and glands, penis, and clitoris. So end bulbs are found in these locations, and they are the receptor for cold. Nerve terminal is in skin and mucosa of oral cavity. We can find it. Okay, that is the receptor for cold, that is nerve terminal in the skin and in the mucosa of oral cavity also we can find it. Next is the Pacinian capsule. Receptors for fine touch located in mucous membrane and pressure sensation, dermis and ligament. So Pacinian capsule, that is a receptor for fine touch located in mucous membrane. Okay, and mucous membrane, you know that that is the inner side of the nasal cavity, is found in oral cavity also. So that is located in that location and is responsible for the pressure sensation, and that is found in the dermis just below the epidermis and in the ligament, which is the what? So what is the ligament is doing? Ligament is connecting bone to bone, and tendon is connecting muscle to bone. So these are found in the ligament means in the we can see that bone to bone connecting joint, that is a say we can say that that is Pacinian capsules are responsible for the pressure at that location. So they are the lamellated elliptical structure. They are large and visible to naked eye, and each capsule is formed of a large number of concentric lamellae of connective tissue fiber lined by single layer of connective tissue cells, and the spaces between the lamellae filled with the fluid or semi-fluid substance. And there is an inner bulb in the center, and each capsule is supplied by a single myelinated naked axon extend through the center of the inner bulb and in a knob-like expansion. Next is the Meissner's capsules. Meissner's capsules, tactile capsules, and they receive the fine touch sensation and found in the dermis of hand, lips, and teeth. So where we can find dermis of hands, lips, and teats, Meissner capsules are found, occur in the hairless portion of the skin, especially most numerous in the fingertips, palm of hand, sole of feet, and they lie within the connective tissue of the dermal papillae. They are oval, composed of flattened, horizontally arranged connective tissue cells and lamellae, and surrounded by the connective tissue capsule. And one myelinated fiber is distributed to each capsule. Then next, muscle spindle. It is a neuromuscular spindle. In voluntary muscle, sensory nerve in end bulbs and muscle spindles are much that the muscle spindle is a cylindrical elongated structure within which are one or several muscle fibers are there. Connective tissue, blood vessels, and myelinated nerve fibers are there. Then muscle tendon spindle, that is organ of Golgi, that is also called, found at the junction of the muscle and tendon. Spindle shaped composed of tendon bundle covered by connective tissue capsule. Next is the mechanoreceptors, is a sensory cell that respond to mechanical pressure or distortion. And there are four type of mechanoreceptors in the hairless mammalian skin. What are the different? Accordingly, we can divide like Pacinian, Meissner's, Merkel nerve ending, and Raffinese capsules. Pacinian capsules are lamellar capsules, which we have discussed in another classification, and Meissner capsule tactile receptor found in the tunica submucosa. Then Merkel nerve ending, that Merkel cell, tactile epithelial cell, oval shaped mechanoreceptor, essential for light touch sensation, found in the skin of vertebrates. Then Raffinese capsule, bulbous capsules, responsible for the substantial pressure on the skin and perception of the heat. So we can see that free nerve endings here, non-capsulated tactile capsules in like appearing like this, encapsulated tactile capsules appearing like this, lamellar capsules are like this. It is a neurotendinous spindle. So like that we can see the shape is different in the different receptors. Okay, vision, rod cell, con cells, hearing cells, smell cells, okay, bipolar cells, taste cells, the free nerve endings, touch and Meissner capsules appearing like this. So there is a variation in the structure also. Raffinese capsules, here we can see Krauss impulse appearing, Presilin capsule appearing like this, free nerve endings are appearing like this, Meissner's disc is appearing like this, hair follicles, nerve endings are appearing like this. So there is a difference. Now conduction across the synapse. So in order for neural control to occur, information must not only be conducted along the nerve cells but must also be transferred from one nerve cell to another across the synapse. Next is the most synapses within the nervous system are chemical synapses involved in the release of the neurotransmitter. Now, the relation between the two neurons are of the nature of contact rather than complete structural continuity, and this junction of two neurons is called synapse. Next, the synapse may be defined as a point of contact of the cell membrane, means plasma membrane of an axon terminal with the axial membrane of the another axon. This is called the synapse. Synapse may be between the axon terminal of one neuron and cell body of the another, means axosomatic synapse. So it may be two types. Okay, synapse may be defined as a point of contact between the cell membrane, means plasma membrane of axon terminal with the cell membrane of another axon, another neuron, and it may be between the axon terminal of one neuron and the cell body of another, and it may be between the axon terminal and dendrites of another neuron. So it is maybe in this way we can divide them accordingly. So we can see this is the axon, this is the mitochondria, this is the synaptic knob, this is the synaptic vesicle, this is the endoplasmic reticulum. Okay, this is the presynaptic membrane, this is the postsynaptic membrane, this is the synaptic cleft. Like this we can see the differences. Now neuroglia. So neuroglia are the another type of cells, that is the two types accordingly. Central nervous system neuroglia are another type, and peripheral nervous system neuroglia are different another type. In the central nervous system, the glial cells are astrocyte, oligodendrocyte forming the sheath we have seen. Microglia and ependymal cell are also there. Then in the peripheral nervous system, Schwann cell or neurolemacytes and stellate cells are there. So we have to see that the central nervous system neuroglia is visible here, that is the ependymal cells are there like this is the spinal cord in the section. So first is the astrocyte. First central nervous system neuroglia is called astrocyte. They create supporting framework for neuron and create blood brain barrier. Astrocytes is the cell which is forming the blood brain barrier. They monitor and regulate interstitial fluid and surrounding the neuron, and they secrete chemical for the embryological neuron formation, and they stimulate the formation of scar tissue secondary to central nervous system injury, and they are the largest and most abundant. So we can see like this is the these all are the astrocytes. So what is they doing? Actually they are forming the blood brain barrier. They are largest and most abundant also. Next is the oligodendroglia. Oligodendroglia that create the myelin sheath around this axon of neuron in the central nervous system. Okay, so oligodendroglia that create the myelin sheath around the axon in the of the neuron. Okay, so what is they are doing? Myelin sheath around the axon of the neuron of the in the central nervous system, and the myelinated axon transmit impulse faster than unmyelinated neurons. Then microglia. So this is the microglia cell which you can see. They are the brain macrophage. They phagocytize cellular waste and pathogens, and microglia derive from mesoderm. Another cells of the central nervous system are derived from ectoderm, but microglia are the specific, or we can see that they are derived from the mesoderm, not from ectoderm. Next is the ependymal cell. They line the ventricles of the brain and central canal of the spinal cord, and they produce and monitor and help to circulate central nervous system or cerebrospinal fluid. And the cell found in the choroid plexus that secrete the central choroid, we can say that CSF, so the cerebrospinal fluid. So this ependymal cell, that is the lining the ventricles. We know that ventricles in the brain, that is the two lateral ventricle, the third ventricle, then fourth ventricle. So these are the ventricles which is lined by internally this kind of cells, and they are central canal of the spinal cord also they are found. They produce, monitor and help the circulate the means produce, help and monitor the cerebrospinal fluid, and the cell found in the choroid plexus that secrete the CSF. So the cells are founding in the choroid plexus secreting the CSF, which is monitored and produced and circulate with the help of this ependymal cell in the cavity or in the ventricles of the brain. Now neuroglial cell of the peripheral nervous system. So there are two type of cells, Schwann cell and satellite cell. Schwann cells, these are the flat cells undergo spiraling around the axon outside the CNS, means inside the PNS. And each axon in the PNS comes with the contact of many Schwann cells and form myelin sheath by depositing concentric layer of lipid and protein. So by depositing lipid and protein, they are forming the covering, and the sheath is interrupted at regular interval in between the covering of the Schwann cell, and this exposed part of axolemma is called nodes of Ranvier, and the Schwann cell are derived from the neural crest cells. Okay, so these are derived from the neural crest cells, we can see that. Now the next thing is the satellite cells. These are the smallest flat cells derived from the neural crest cells. They are also derived from neural crest cells and form the capsule around each ganglionic cells. And some oligodendrocyte are also serve the purpose of satellite cells around the cells of the gray matter. Okay, so like this in the peripheral nervous system, there are two types of Schwann cells in the neuroglia, or neuroglial cells, satellite cells and Schwann cells. Then how the regeneration and repair occurring? So cell body dies and no regeneration occur if cell body dies. If axon fiber damage, Valerian degeneration, that is called, and loss of nerve distal to injury, that is also possible. Chromatolysis means change in the nerve cell body. Then neuropil refer to the axon, terminal branches, dendrites and glial process that collectively form a background matrix for the cell body seen within the light microscopy. That what is the neuropil now? So it refers to the axon and nerve terminals, dendrites or glial process that collectively form a background matrix for the cell bodies seen in the light microscopy, that is called neuropil. And gray matter of the surface of the cerebellum and cerebrum is called cortex. So gray matter is in the cortex, in the cerebellum, means gray matter of the surface of the cerebellum and cerebrum is called cortex, and distinctive gray matter masses within the central nervous system are usually designed at nuclei. And nervous tissue exhibit a variety of configuration among the different region of the central nervous system. And the characteristic neuron of the cerebral cortex has a pyramid shaped cell body and oriented with its apex directed towards the surface. Then dendrite emerges from the apex and basal edge of the pyramidal cell, and the axon leaves the center of the base and enter the white matter. So now the cerebrum in mammal, but all but the ventral cerebral cortex is designed neocortex because it is phylogenetically recent, and cerebral neocortex is divisible into 6 layers, although the layering is evident only in thick section and the prominence of individual layer varies from region to region. So like this we have to see that different layers of the cerebrum, and the cerebrum in detail we will discuss in the next class. So we will stop here up to the cells and the regeneration of the central and peripheral nervous system. Thank you.